like to thank Lee and Kathy and the other members of the selection committee for nominating me, whether I deserve it or not. Of course, I'd also like to thank very much Michelle Smith and Laurie and Michelle Darman and the other staff at Podium Conferences for making all the arrangements and generally treating my wife Connie and I like royalty. Greatly appreciate it. And lastly, of course, I'd like to thank you all very much for sticking to the bitter end of a long conference. I know if I had my choice, I wouldn't be here. I'd be out somewhere looking for a sake bar. And so, as Lee said, you know, when I first got the message from Kathy last fall saying that I would nominate for the summer, I'm sure they made a mistake. And I suspect they actually meant to send it to John Krakauer, but they clicked on my name by mistake on the mailing list, and so here we are. Lee insisted I wasn't there. So I'm both very surprised and flattered and very honored by this reward. And also, I have to confess, rather uncomfortable. I mean, these are photos, as I understand it, of the people who received the award over the years. I mean, these are really outstanding people which we received. Yeah. You know, I would never be so delusional or have the hubris to consider myself in a league of people like Peter or Roger and Grady, for heaven's sake, I don't belong in the crowd. So, and the other reason I feel a little bit uncomfortable, you know, I quickly put together this list, where at least 50 people really should be up here talking steadily. And I'm going to pass this on to Lee and Kathy so they don't make this mistake again in the future. But again, the other reason I feel uncomfortable is that, you know, unlike all these other outstanding scientists, just about everything I've read in our field over the last several years seems to imply, if not states explicitly, that almost everything I've ever done in my career and others of my ilk has been naive, simplistic, and misguided. And I'll try and explain why as we go along. So anyway, as a result of all that, I think this is probably the best representation of how I feel. I don't know how many non-North Americans get this cultural reference, but anyway, that's where we are. So I'm going to start talking. I thought, for my keynote address, I don't want to drone on too much about our work. I think it'd be interesting for, you know, especially for the youngsters in the audience, like Ed Sullivan used to say when he introduced Topo Gijo, you know, to sort of provide a bit of a more historic perspective of evolution of research in our field, and maybe somehow describe a little bit about what we did and how it reflected thought, thinking in the field, and maybe contributed to it. So let's start with the golden era of cortical neural recordings of long-term movement, sometime in the mid-1960s up until the early 80s. Studies of cortical control movement were involved in maybe single-unit recording and single molecular recordings that were usually focused on identified pyramidal neurons, pyramidal tract neurons. The control of voluntary movement at that time was assumed to be best understood by reducing it to the simplest element, rotations about a single joint. And control of more natural multi-articular movements therefore could be approximated by a linear sum of the component single joint actions. Of course, everyone knew that was a simplification, but they thought that was the best way to get a handle of things, to start with single joint rotations. We didn't have many computational models at that time, and the closest thing to it perhaps is the transcortical control loop model of motor control. The idea that the primary motor cortex occupies the pinnacle of a series of fast feedback loops that, amongst other things, serve to defend stable postures and movements against external perturbations. And despite the limitations of technology, the limitations in conceptual understanding motor control, all these pioneers made fundamental contributions and provided experimental, empirical evidence that is still valid to this day. For instance, LR showed, of course, how single neurons in the motor cortex responded during movements to a particular part of the body, and more specifically recorded and responded to particular rotations of single joints. In his second seminal study, he showed how he addressed the muscle movement dichotomy that had been around since the discovery of motor cortex in the 19th century. And he showed how single neurons Many pyramidal tract neurons, when the movements are unloaded, would increase their activity if a load resisted movement, and others when they diminish their activity when the load assisted the movement, suggesting there's kind of a muscle-like property to the responses. Of course, he also found that many of the neurons didn't behave that way. They did other things that didn't make a lot of sense. Other people like Fetz, Cheney, Rajalina, and others looked in more detail at the properties of pyramidal tract neurons, and then finally at some point, Fetz and Cheney discovered the existence of these neurons was monosynaptic connections to spinal motor neurons, the so-called corticomotor neuronal cells, or CM cells. And so of all the cells in the brain, these should have the closest relationship to muscle activity, and yet they found that it didn't. Here are the histograms of CM cells and their target muscles. They didn't correspond cleanly to what the muscles were doing. And sometimes they get very weird responses. Here, a later study by Griffin and Cheney. You see this CM cell with multiple bursts. Some of them seem to correlate with some of the bursts of some of their target muscles, and didn't seem to correspond to the activity of the target muscles at all. So even pyramidal tract neurons, the single monosynaptic connections of our neurons, did not look like muscles. Thatch published a paper in 1978, which he trained monkeys to make stepwise reciprocal movements of the wrist against assisting and resisting loads. And he tested the activity of the neurons against 3 predictor models, one that looked like a muscle, one that tracked the position of the joints, and one that predicted the direction of the next movement in the sequence. And he found a broad range of activity in the motor cortex, including cells that look muscle-like, joint-like, and predicted the next direction. And more problematically, many of the neurons show partial correlations to all of these different models. So what are these neurons coding in their activity? And one last paper I'm going to mention is a paper by Humphrey, Schmidt, and Thompson, published in 1970. He also did an Everts-like paradigm, single joint rotation, but the innovation he made, he actually inserted 5 independently positional microelectrodes in the motor cortex and recorded from small sets of single neurons simultaneously while the monkey was working in a task. And he found that despite the simplicity of his task, here 5 neurons occurred simultaneously. They showed widely divergent patterns of activity when the monkey was making these simple movements. And he also showed that you could combine the activity of these single neurons in certain ways, and the more neurons you added, here's first neuron number 5, then 3 to 5, and 1 to 5, you add them together, you can find better correlations to certain motor output parameters. So they anticipated the idea of population coding and decoder algorithms in 1970. And they also found that, again, when you look at these combinations of neurons, they show partial correlations to most of the parameters they measured: displacement, position, velocity, force, and derivatives of force. So, what are these neurons contributing? I think, I don't know if any of you have ever read that paper. If you've not read that paper, you really should. But these are some of the quotes from that paper. We present data which suggests that information about a given movement is carried not simply in the discharge pattern or spike trains of individual cortical neurons, but to a significant extent by the temporal relationships between them. By simply weighting and summing the discharge frequencies of each cell in the set, we're able to predict the time course of certain response measurements with unexpected accuracy. We take such findings to suggest that the temporal relationships between the spike trains of separate cortical cells may be a significant variable in the control of movement, and as important, if not more so, the particular type of firing pattern exhibited by any given neuron. Thus, our data allow no simple answer to the question of response variables most highly related to and therefore possibly controlled by the activity of cortical neurons. Instead, our results suggest that answers to such questions may well depend on whether or not the neurons are there simultaneously, so that important temporal relationships between these discharge patterns may be taken into account. But these were really prescient observations and interpretations. Now, in 1970, to many in the audience, that's prehistory, right? I mean, it's certainly prenatal for these pathways. But nonetheless, this shows that there were very smart people working in the field, despite what you might think where they were doing very simplistic experiments. And they had already identified issues that have bedeviled the field ever since. And that they, I lost my train of thought here. They recognized that there are things going on in the motor cortex that they didn't have a handle on. So very prescient observations way back. So it was about this time, and in that behavioral, in that historical context, that I joined the Apostles Lab. Actually, I was originally recruited by Vernon Mountcastle, but a couple months after I started, we were chatting, and he said, well, you know, how are things going? Blah, blah, blah. I said, it's very interesting, but you know, you're doing all these visual stuff in Area 7 I really wasn't interested in. He says, well, why don't you go talk to the Apostles for a job with us? He's just setting up his lab. He's doing arm reaching and parietal cortex and so on. You may be more interested. And we started talking. And sure enough, we hit it off, and I made the switch. That was very kind of him, but I suspect in reality he just wanted to make room for Richard Anderson, who was coming into his lab a couple months later. So, anyway, I ended up with Apostles for that. And we started training monkeys in this reaching task. Now, many of you probably don't know that the original objective of this study was actually to follow up on Vernon Mountcastle's classic 1975 paper on command neurons in the parietal cortex. We were going to record in Area 5 and Area 7 and look for command neurons. But Apostle decided it would be a good idea to record a few neurons in motor cortex, sort of give a benchmark, get a handle for what goes on in motor cortex, and then we go back to the parietal cortex and pursue our project. But it ended up that what we found was so interesting that we never left the motor cortex, and the parietal project became the back of our project. It's also interesting that at that time, a lot of the people thought we were quite mad. You know, they couldn't figure out how to control single rotations or a single joint. They figured that you'd never make any sense out of what these neurons are doing while the monkey's just flailing his arm around in space. But, you know, that didn't deter us. We went ahead and did this experiment. And sure enough, it actually was much more simple than we thought. And then again, we showed this broad continuous attuned activity, not chaotic patterns, reciprocal in certain axes of movement, like Edwards had reported. And the single neuron activity turned out to be surprisingly simple in this paradigm, unexpectedly so. And I often like to say that this figure itself is actually one of my main contributions to neuroscience, because I made it. It was my first exercise in programming in Fortran 11, PDP 11 Fortran. And so my job was to print out all the rasters of the neurons in motor cortex that we collected, and I thought the circular arrangement was particularly elegant. And so that's how that figure came to be. And in fact, if you're interested, I'll be signing copies of the figure at the lobby. $25 a piece, if you don't mind. US, of course. And we also found that many neurons, again, muscles show the same thing. And so these are actually data from one of my later studies, but that's irrelevant. We had found that the muscles in our task show the same broad directional tuning. So we wrote this up in the results section of the original version of our manuscript that we submitted to the journal. And we had a section in the discussion talking about the implications of this similarity of the tuning properties in neurons and muscles, and how this might imply, what this implied about control of muscles and muscle synergies from the motor cortex. But the editor, who I'm quite sure is Ed Edwards himself, told us to take it out. He said, you never recorded from identical primal tract neurons, therefore you have no right to speculate about the nature of the sending motor command from the motor cortex to the spinal cord. So we pulled that out. I've often wondered how people would have changed their interpretation of our paper if that section had been left in. So the other major finding, of course, we made is that many cells are broadly tuned, different cells have different preferred directions. The entire direction spectrum was represented in a fairly uniform fashion. So that was a problem. The pattern of activity during a given movement is diverse, complicated, distributed all over the arm motor map. How can an observer, such as the spinal cord, make any sense of this? If he receives some kind of unbiased sample of this activity. Or putting it another way, could we extract from this complicated, diverse pattern of activity a signal that predicted what the animal had done, or which direction of movement he had made on the basis of the activity? So again, as you all know, we decided to do something very trivially simple. We represented each neuron by a vector, aligned to its preferred direction. We scale the activity, this is the vector, as the length of the vector as a function of activity observed in each direction of movement. We threw all that in a pot, added it up linearly, and came up with this result. Again, this is my other major contribution in neuroscience, because I also made that figure. This simple, trivial exercise actually work much better than our wildest expectations. The summed activity of that population pointed in a direction that corresponded surprisingly well to the direction of moving the animal had made. So we had a clear signal that allowed us to say what the animal had done in response to each of these different patterns of activity. And so arguably this population vector is arguably the first large-scale neural decoder that eventually been used in many other applications. And this phenomenon has been replicated many times. This is a result of my first experiments in Montreal. Apostolus, Andy Schwartz, and Mark Kentner extended it to three-dimensional pointing and also extended it into time by calculating the instantaneous population vector between milliseconds and gluing them to detail. You can see that the neural trajectories of broken lines correspond surprisingly quite well to the actual projections of each of the tasks. And Andy Schwartz extended this to more complicated geometric tracing results, and he showed again that these instantaneous population vectors would predict the motor output some 100, 150 milliseconds into the future. So these various analyses show that the motor cortex is a rich source of signals about the intended actions of the subject. And as I said, the population vector algorithm was later shown, its utility was confirmed with many of the initial brain-machine interface studies by John Donahue and Andy Schwartz and others. But the population vector model has perhaps fallen somewhat out of favor, for some reason perfectly good. Far more sophisticated decoder algorithms have been developed that extract much more efficiently the motor intention information available in the motor cortex. But I think it's also fallen out of favor for a wrong reason. And that's because in the minds of many people, it seems to have become indivisibly linked to the hypothesis that motor cortex is coding the spatial displacement of the hand during reaching movements. And I think that that's a big mistake, as I'll show you. A population vector and hand-space kinematics are two completely different things that are easily dissociable by experiments. And that's what we're trying to show you in some other experiments. But at the time that we were doing these experiments, again, the way in which we were looking at motor control was being reframed. Rather than single isolated movements or single joints or focusing on primal tract neurons, they started to look at it in a broader perspective as shown by this next figure. Here's a completely spontaneous, unposed photo of me contemplating a delicious Montreal breakfast. I think about counting about 150 photos to get two that are reproducible. Here you see, proud McGill dad now. I've been a professor at the University of Montreal for 30-something years. That's because I've never been good enough to be a professor at McGill, but at least our daughter made it, so I'm proud of her. So, they started to get motor control on the basis of two broader questions. How does the motor system get the skeletal motor plant from some initial state to some desired final state? And a related question is, how does the motor system, how does the brain bring together many different central and sensory signals in different kind of coordinate frameworks and generate a motor output in the coordinates of muscles and muscle activity? So this seemed to make sense. It seemed to immediately become a question of sensory motor coordinate transformations. And at the same time as these ideas were evolving, people were doing psychophysical studies, started looking for invariances in motor control that presumably reflected the kinds of computations that the brain was doing to produce, to translate from the initial state to the final state. That bagel and smoked salmon was delicious that day. For instance, Pietro Morasso showed that typically reaching movements from any particular start point, any end point, it was generally linear, approximately linear, and showed a symmetric belt of bell-shaped velocity curves. independent of the complexity of the joint rotations required to produce those different movements. So it had simple properties and expressed in spatial coordinates. The velocity curves scale very nicely with distance and the acceleration curves. And these invariances even seem to be almost effector independent at some level. This is a study by Mark Raber. This is an example of someone writing this phrase using the right hand as normally or with the wrist fixed, writing with their left hand, writing with their pencil and head and their teeth, and even clenched between the toes of their feet. So there were complex trajectories that represent the writing style of this subject was clear and repeated despite the fact he was using very different effectors. So somewhere in the brain there must be some kind of representation of these effector-independent representations of these actions. Other studies looked at and did analyses of endpoint errors. For instance, Gordon and Guez and Ghilardi found that the variability of errors in distance and amplitude were independent, suggesting that maybe these two properties of movement were planned by separate channels. Others looked at the distributions of endpoints during reaching movements and suggested that errors in the computations and noise in these various transformations could explain the endpoint errors in different coordinate frameworks. This is an example of one from John Seckley and Martha Flanders. They said that reaching movements to different targets and different spatial locations and distances could be accounted for by transformations in these coordinate frameworks. So many lines of convergent evidence suggested that maybe the brain was performing something that looked like a sequence of coordinate transformations between representation of movements in different coordinates. And the heuristic value of this model, obviously, of course, is that it makes predictions about what kind of response properties we could find in the brain that if neurons are performing these different computations representing motor output in these different frameworks. And so this hypothesis dominated the field for many years. I use this myself very often, obviously, for designing experiments that look for evidence of what kinds of information neurons are processing in parts of the brain. For instance, my first day I went to Montreal wasn't very creative. I just revisited Edouard's old muscle movement paradigm and took it into 3 dimensions, in reaching movements in 2-dimensional space. I should point out that all the data I'll show now from our experiments are collected in the primary motor cortex, in area 5, in my PD parietal region, and in the dorsal premotor cortex between the jejunum of the arcuate and the precentral dimple. I also want to emphasize that almost all the data I'll show you are collected in the bank of the central sulcus, what Peter Strick calls the new motor cortex, where all the CM cells are found. Where many of you put your microelectrode arrays on the crown of the sulcus is what Peter calls the old motor cortex. And my experience is that these are 2 slightly different beasts. The data I'm going to show you are from the bank of the central sulcus. We train monkeys to make reaching moves in 8 different directions with 8 different external loads. Pulling in the arm, this is a muscle activity, as you see here, nicely directionally tuned. You can represent that by a polar plot. The other polar plots show how the muscle activity increases when the loads resist its action, decreases when the loads assist its action. So, basically the muscle is doing its job, generating forces and torques required to compensate for external loads and move the arm in different directions. Here, 2 other muscles. So this is a nice signature of what a peripheral force generator looks like in these task conditions. Here's a primary motor cortex, broadly directionally tuned. Now we apply a load that pulls the arm in the direction opposite to the preferred direction of the neuron, somewhere up here. Just as Edouard showed, the cell becomes much more active when the load assisted the activity of the direction of movement of the cell. It got less active. When we tested across all 8 directions, you see this beautiful signature again, continuous modulation as a function of direction of movement and as a function of these external loads. Very similar again to what muscles look like. So many cells in the bank of the central sulcus have properties that resemble the muscles in these task conditions. conditions. But many others show less sensitivity to external forces while still being very strongly direction tuned for reach movements. More of these more kinetic neurons are in the central sulcus. Cells that are at sort of this end of the range tend to be more on the gyrus, the crown of the gyrus. In 2000, Emo Todorov published a study in which he did some simulations of what motor cortex neurons would look like if they directly controlled the contractile activity of single muscles or muscle synergies. And he represented these results in terms of the population vectors. And he showed, of course, that when the monkey's making reaching movements without external loads, the population vectors tend to point in the direction of movement. When he also calculated the changes in activity, while the monkey is compensating for these external loads in eight directions. I want you to notice something neat or interesting. The vectors are no longer just pointing straight into the center. There are clearly some anisotrophies. They're longer for lateral loads than for loads that pull the arm toward and away from the body. And these vectors are deviated away from the pure 45-degree angles. This was explained as modeled by the anisotropic properties of the arm in a simulation, the compliance and stiffness of the arm. The arm is a lever, and he's looking at shoulder-related neurons, and they have to work a lot harder when they're moving, kind of dealing with loads this way than this way. It also showed what would happen when that population of neurons makes reaching movements to the left with constant kinematics while compensating for these different loads. Basically, it's just a simple vectorial sum of the movement-related vector and the load-related changes in activity. You see clearly that the population vectors change dramatically as a function of external loads and clearly are deviated away from the actual direction that the animal is moving his arm. And I don't want to brag, but that's exactly what we found in primary motor cortex. The population vectors showed all these evidence of pointing direction of movement, these clear anisotrophies that we presume reflect the biomechanical properties of the limb, and these deviations of activity away from the kinematics of movement while the monkey is making movements to the left with constant kinematics. Now, these findings are not trivial. If you go to other parts of the brain, they don't show these properties. We recorded from shoulder-related neurons in the parietal cortex, broadly tuned for direction of movement, as you see here. But when you test that cell with the external load, it's completely oblivious. It doesn't seem to show any knowledge that he's actually working harder or dealing with external loads or not. That was typical. This is with a range of load sensitivity we saw in area 5. And interestingly, unlike area motor cortex, when neurons did show some modulation of functional loads, you put them all together, that load sensitivity disappears. They all seem to cancel load at the population level. So area 5 was really working hard not to show evidence of the kinetics of the task. It seems to be trying to represent information about the spatial temporal form of the movement. Again, this is shown clearly when you do this population vector analysis of the activity in area 5. When the monkey is making one direction movement with all the different load conditions, I want to assure you it's not the same figure just copied 9 times. It's really the real data of the neurons under these different load conditions. And it's strikingly different compared to what we see in primary motor cortex. So this doesn't prove, of course, that area 5 is trying to code the kinematic parameters of movement, and motor cortex is trying to code the kinetics of the output. But it does show clearly that the information being expressed in area 5 captures something about the spatial temporal form of the movements, while activity in primary motor cortex is also expressing information about the underlying causal kinetics. These were analyses done by averaging activity across the entire duration of movement. In a study done by Laura Sergio when she was a postdoc in my lab, we wanted to start looking more at the temporal dynamics of this. So we trained the monkey in 2 different tasks. One in which he generated horizontal ramps of force in 8 directions in a horizontal plane, and also when he made movements in a horizontal plane while pulling a heavily weighted handle back and forth. So now he's dealing with inertial loads. I won't go into all the details of this paradigm, but it was very hard to train the animals, and Lauren did an absolutely spectacular job. training these animals to be very skilled in the paradigm and collecting the data from these experiments, very challenging experiments. This is an example of a muscle, posterior deltoid, in the isometric task and in the movement task. The lines here show the forces measured at the hand in the two tasks, and we see the histograms of the contractility of the muscle in the task. Of course, in the isometric task, you see these beautiful ramps of isometric force in 8 different directions, and continuously modulated ramps of changes of activity of the muscle. In the movement task, the force profile is much more complicated. The monkey had to push on the handle to accelerate it. Then halfway through the movement, he had to actually pull on it to slow it down. And then he continued to push on it to hold that heavily weighted handle against gravity over the targets. So you get this complicated waveform of output forces in order to perform the task, while moving in these beautiful, smooth hand trajectories. And of course, the muscle shows that same parallel behavior, this classic triphasic response behavior. Burst, pause, step, and silence, burst, silence. So the muscles, again, are just doing their job. They're generating the forces necessary to produce these motor outputs. There's a second muscle with the same responses, continuously modulated ramps of activity in the isometric task, the triphasic response pattern in the movement task. Now, if you represent the activity of this muscle at 20 millisecond intervals in an isometric task, you see this continuously tuned direction, activity in the same direction from the start of the trial toward the end of the trial. In contrast, when you do that same exercise with the activity in this movement task, you see it starts to go in the same direction, but suddenly whips around by about 90 degrees and then returns back to its original direction. I mean, this is not some random flop. This is a change in directionality that captures how the activity of that muscle changes at different times to serve either as an agonist or as an antagonist during movements at different times in different directions. So this is a meaningful change in directionality of the neuron during movement, of the muscle during movement. This is a motor cortex cell in the same paradigm. See the nice tonic changes in activity in the isometric task with a little bit of a phasic burst at the onset. And this far more complicated triphasic response pattern during the movement paradigm. There's a nice triphasic burst there. When we did the instantaneous tuning of the cell, you see that the continuously tuning isometric task starts off in the same direction in the movement task, then flips over in the approximately opposite direction, and then reverts back to its original direction. So that neuron again is paralleling the dynamics, the kinetics of the task in time. Here's another neuron showing the same properties, monotonic changes in tonic activity in the isometric task, more triphasic response pattern in the movement task. And if we take all the neurons aligned up their tuning curves to the right, the thin line shows the activity, the average activity of neurons in motor cortex in the isometric paradigm. The thick lines show the behavior in the movement paradigm. You see these extra components of activity, burst, pause, step, and delayed burst in the opposite direction at the level of population. You can capture that again with a nice vector analysis. This is the vectors of instantaneous forces in the isometric task, and the monkey's pushing to the left and to the right. This nice ramp change. This is the activity of the population of the neurons in the isometric task, consistently pointing in the direction of the isometric forces. In contrast, in the movement paradigm, you see an initial agonist force in the direction of movement, then a decelerative force in the middle of the movement, and then a step to hold the handle over the targets. And obviously, the instantaneous population vector analysis shows very similar parallels that lead the output force by about 150 milliseconds. So again, motor cortex activity is generating activity that is reflecting the temporal dynamics of the different tasks. And you can show this in a different way. Here, the hand paths in the two parent dishes, where there's no hand path, no movement in the isometric task. Nice, smooth, displaces the hand to the target in the reach paradigm. When we glue all of the instantaneous isometric vectors tip to tail, see this beautiful force aligned in the direction of the targets. In the force trajectories during the reach movement, though, it starts to go in the direction of movement. Then you see a little curly cue while the monkey's trying to slow down the movement. And then again, this step to hold the target over the target. And the neural trajectories, again, they're fairly smooth. Now they're strongly biased away from the actual direction of displacement of the cursor. Again, we suspect this reflects the anisotropic properties of the shoulder or the limb. And again, and here you see these curly cues and jiggles or whatever. So the primary motor cortex, again, is expressing information that captures details about the differences in the dynamics of the force input in these two paradigms. And again, this is highly relevant because if you do the same exercise with the parietal cortex, you don't see anything. The cells are strongly active in the reach task without any sign of that triphasic response. And strikingly, in the isometric paradigm, there's hardly any activity in area 5 because the limb is not moving. This shows the population activity in area 5 in the preferred direction, all the neurons in the opposite direction. It's completely missing all these complicated patterns that we see in primary motor cortex. And when you do a population analysis, population vector analysis, you see clearly the area 5 activity is consistently pointing in the direction in which the animal is moving his arm throughout the entire exercise. But the motor cortex shows us an initial push in the direction of movement, then a reversal while the animal is trying to break the movement, and then return to the direction which is holding the handle. So clearly this activity shows that there's a striking difference in the temporal dynamics of activity in area 5 compared to primary motor cortex. So these are consistent with the predictions of the sensory motor cortex transformation model that says that some cells somewhere should be mainly concerned with the spatial temporal form of the movement, while others should be concerned with the outward forces. Another prediction of the model is that some cells should be more concerned with extrinsic properties of the movement, other cells should be more concerned with intrinsic properties of the movement, intrinsic to the arm. And that was the objective of Steve Scott's experiments we did in postdoc in my lab. In fact, the real reason for the experiment, again, was to try to counter the misinterpretation of the 1985 paper that neurons and motor cortex are coding the head-space kinematics of movements. The animal is trained to make movements along the same spatial trajectories while holding its arm in two different postures, a natural paracentral posture and one in which the arm is abducted up into the horizontal plane, shown here. Clearly, the two movements require different joint rotations, different muscle activity, and so on, while still displacing the hand along the same spatial trajectory. We found that many cells showed strong differences in their activity, either its overall level of activity and in particular its direction of tuning, when the animals were performing the movements in the same spatial trajectories while holding the arm in two different postures. So right away, this shows clearly that the spatial tuning of these neurons, single neurons, is not explicitly signaling where the hand is going in space. It's signaling something else. Another clue is that when we looked at the preferred directional tuning of neurons related primarily to the elbow versus those in the shoulder, again we see differences. The elbow-related neurons are mainly active in this axis, of course, of which the main configuration of the elbow in both planes. And shoulder cells are more uniformly distributed in the natural posture and start to show some evidence of a bias when the animal is holding his arm up in the abducted plane. These differences presumably reflect again the different degrees of rotation of the shoulder versus the elbow. Another evidence of that this is capturing something about the intrinsic biomechanics of the movements is that we separately plotted the activity of neurons recorded in the two separate hemispheres while the animal is moving the arm contralateral. And you look at the shoulder-related neurons in this task, and you can see that this bias in activity in the animals moving in the abducted posture is mirror symmetric. You can capture the mirror symmetric intrinsic biomechanics of the two arms. Another thing we found that was quite interesting is that this is a scatter plot of the preferred directions of the cells in the natural posture versus the changes in activity when he rotated his arm in the abducted posture. It was not random. There seemed to be some kind of structure. to it. And we thought this was an important clue, but Steve and I were talking about these experiments one day, and we were thinking, it would be really cool if we had some good models that made strong predictions about what neurons would do in this paradigm if they were coding information in different kinds of coordinate frameworks. And I swear that 2 days later, Steve came back and he said, I've worked out the models and here's the results. He first showed a model in which neurons, simulated neurons, coded the extrinsic spatial kinematics of the movement, and clearly they're not at all sensitive to the difference in posture, because they're oblivious to that, right? They only care about what the hand is doing. So we could reject categorically that the motor cortex neurons were behaving like hand-space kinematics neurons. A second model was a joint-centered kinematics model. And here we start to see things that resonate with our actual data. Look at the scatter plot of the changes in direction and directional tuning of the neurons, of these simulated neurons. It looks very similar to our actual data. Look at the distributions of preferred directions of the simulated joint-centered neurons related to elbow rotations and shoulder. Very strongly biased here. More uniform here with some bias in the abductive plane. And again, that seems to really resonate with what we actually saw in our data. Finally, Steve developed a joint-centered torque model. And clearly, this is far more structured than our actual neural data. And the distribution of preferred directions of these joint torque neurons is far more biased than our observed neural activity. But intriguingly, they look much more like what the muscles do in the paradigm. So muscles are generating torques, and this torque-centered model showed the parallel what happens to the muscle activity in the task. Right? You know, when Steve, to my mind, these models really made the study, because it really gave us clear predictions of what different neurons would do if they're nominally coding information in different coordinate frameworks. And I have to say that when Steve showed me these models, I was really impressed. And I said, this guy's going to do all right. I think he's backed up that prediction well. So what are the implications of this finding? You know, what we see here is that sensory signals related to the posture, the starting posture of the arm, while the monkey is holding his arm in either a natural or an abductive posture, has a strong effect on the directional tuning of the neurons. And basically, it puts the motor cortex into two different initial states that we believe facilitates the generation of the motor commands required to make those same movements in the same spatial trajectories while holding the arm in two different postures. Now, I don't know whether the idea of initial states that lead to different neural output trajectories resonates with anybody in this audience. But I think that these experiments were consistent with that idea. That changes in posture cause changes in the initial state of the motor cortex, which undoubtedly facilitate the generation of the appropriate motor commands needed to make these movements. And again, this is not a trivial observation, because when we recorded the premotor cortex, they were much less sensitive to changes in arm posture. So clearly they're doing something different than the motor cortex cells. And again, this is consistent with the sensory motor coordinate transformation model that says that somewhere in the brain there should be cells that are concerned more about what's extrinsic properties of the movement, and other cells should be more concerned about intrinsic properties of the movement. Finally, a final example. As I showed, some psychophysics experiments and the model itself said that somewhere there should exist effector independent representations of motor output, while other cells should be dealing with the effector dependent details. And we found, I believe, compelling evidence for that distinction entirely by accident. Most of the discoveries that were made were accidental. One day, Donald Cramlin and I were recording a neuron and premotor cortex. So this may be it, I don't recall. beautifully directionally tuned during the delay period in this task, which was actually designed for completely other reasons. But we were recording this neuron, beautifully directionally tuned. We were just shooting the breeze. Well, what does this mean? What's going on? What does this neuron think it's doing? And we started kicking around ideas about how can we ask this neuron what you think you're doing. And it turned out that this animal had already been trained with his other arm, and we collected data from the other hemispheres. So just, what the heck? Let's see if he can still do the task with what is now his ipsilateral arm, ipsilateral for the wrist recording, and what does the neuron do? And this is what we found. The neuron was active when the monkey was using his ipsilateral arm, and even more importantly, the directional tuning of that cell was very similar to what we saw when he was using his contralateral arm. So this cell didn't seem to care what arm the monkey was using. It just said, I want the arm, or the effector to go over here. I want to reach in this direction here. Completely independent of what arm it used and the biomechanical details of the movement itself. Here's another example from Donald's experiments. Again, very similar directional tuning. Everybody was less active. They're not identical. I'm not claiming that they're identical in the animals using the two arms, but they're the most clear overarching properties is similar directional tuning when he's using either the ipsilateral or contralateral arm. Here's an example from Toyama's experiments. Again, designed to do completely different things, look at completely different issues. But we saw the same property. It's very similar directional tuning where the animal is using the ipsilateral contralateral arm. And again, when you go into motor cortex, it's a completely different story. The cells are barely active in the delay period. They're strongly active in directionally tuned. The monkey's using his contralateral arm to make different reaching movements, and much less active, in this case, completely inactive when he's using the ipsilateral arm. So motor cortex is dealing with what the contralateral arm is doing. Premotor cortex is generating signals that seem to have something to do with what the overall intention is, where you're going to reach into space, independent of the details and biomechanical details, and even independent of what arm the animal is using. So these kinds of experiments we did seem to show some agreement with these different predictions of the sensory motor cortex transformation model. So maybe that means that when I'm having my breakfast, my brain is a sequential sensory coordination transformer that allows me to get my hand, to reach the coffee cup and bring it up to my mouth so I can pick some of that delicious coffee. But as you all know, that model has fallen out of favor for a variety of reasons. And new models have been pushing it aside. For instance, maybe my brain is an optical feedback controller. Still, for the life of me, despite all the attempts of Steve to explain what a control policy is, I don't get it. But this is a model that has had a lot of attention. It was originally proposed by Montreal and Michael Jordan. And of course, Steve Scott and his colleagues have done some beautiful experiments over the last few years to test and promote this idea of optical feedback control in underlying voluntary movements. And of course, as all of you know, the latest flavor of the month model is the idea of the brain as a dynamical network. I don't have to explain to the sophisticated audience what that means, but it suffices to say that it implies that the best way to understand how the brain is working is not to look at what a given neuron is doing at any given moment in time. Instead, it's better to analyze the shared patterns of covariance, of interactions between populations of neurons, and that'll give us a better clue about what the synaptic mechanisms are that are determining the properties of the neurons and the neural networks. And that idea has been aided or in conjunction with these very powerful dimension reduction algorithms that allow us to collapse the pooled activity of many neurons in this n-dimensional activity space into a set of lower level dimensions, a smaller number of lower dimensions that, again, attempt to capture something about the underlying computational dynamics within the neural circuits. And those studies have come to a number of very intriguing conclusions, very interesting clues about what's going on. You know, much of this work, of course, is due to Krishna Shenoy, Mark Churchland, John Cunningham, Myron Yu, Aaron Batista, Hans Scherberger, Mark Schieber. Sorry if I don't list your personal name just for the sake of time. Many people have been making some important contributions to this concept in recent years. For instance, they've shown that in a simple instructive delay task, the neural activity converges on some sort of preparatory state in this low-dimensional activity space, and then eventually evolves outward along a neural trajectory that reflects the motor output. Different motor outputs have different neural trajectories in this activity space. Different movements also have different preparatory states in this neural activity space. In some dimensions of this neural state space, there's evidence of rotational dynamics during movement. And obviously, Mark Churchland can contest that this is surely the most contentious finding of this basic experiments. Other experiments, of course, show that we can consider that there's a limited manifold, an intrinsic manifold of activity in a given task. The neural activity does not explore the entire n-dimensional activity space. And in some regions of that manifold, there are output null regions that do not generate muscle activity, and output potent regions that can generate muscle activity. And so this may explain why there's no muscle activity during delay periods in instructive delay paradigms. Elsewhere, Aaron Batista, Myron Yu, Steve Chase, and others have used these concepts and the idea of an intrinsic manifold to shed very intriguing new insights into how we learn motor skills, how the motor cortex affects motor skills. And for me personally, I think one of the most novel and important revelations of these analyses is that not all activity in motor cortex is concerned with specifying the details of motor output. That there's a lot of activity in motor cortex in these tasks that does not reflect the motor intentions, but rather deals with internal computations, such as the transition between output null and output potent spaces prior to the movement initiation. As Mark Churchland has shown beautifully, it may help to avoid tangling of the neural trajectories for different movements, and as he showed spectacularly the other day, it may also help us to trace the evolving sequence of movements in a cyclic task. So I find that's a very fascinating idea. But this is about me, right? Not about everybody else. And I could go on and praise these wonderful new experiences and new ideas. But what's this got to do for me? You know, what has this got to do for my legacy, if you will? As I said at the beginning, many of these experiments seem to suggest that what I've been doing for most of my career was fundamentally naive, simplistic, and silly. And so, where's my legacy? And I think we can find some of the clues of the answer to that question if you just look at some of the quotes from some of the papers published recently. And many of the authors are in the room, by the way. And you know who you are. For instance, initially such studies seem to provide supported evidence for plausible notions, and not even hypotheses. Our ideas didn't deserve to be called hypotheses, they're just plausible. But in the end, they can just distract us from really getting to the truth of what's going on. Another paper said that representational models lack the explanatory power to describe complex aspects of single neuron population activity. So it just wasn't good. Another paper rather dismissively said about single neurons, well, of course, there'll always be correlations that are incidental rather than fundamental. Right? And another paper said these complex, often puzzling responses of individual neurons, I don't think they're all that puzzling as I tried to show it. can be explained naturally as minor elements in an uncoordinated underlying dynamical system. You know, how to make a guy feel good about his relationship. And then this final quote is particularly hurtful, because it was written by someone I thought was a friend. Neurons thus likely provide only randomly oriented, low-dimensional glimpses of the overall neural picture. The neural analog of blind men examining an eye. But that's a bit harsh. So, in my defense, let me make a few. I should point out that, you know, this same person earlier in his career did the same silly experiments I've been accused of doing. So evidently he's had an epiphany, and as we all know, the newly converted are the most zealous people in defending their newfound faith. So that might explain that. So anyway, let me make a few comments in response to these ego-destroying quotes that I just cited. You know, the correlations of single neurons with particular arbitrarily chosen motor output parameters may very well be coincidental from a causal and mechanistic point of view. Okay, I'll accept that. That makes sense. We all knew that the sensory motor coordinate transformation model was not easily implementable in neurons, right? It doesn't make sense that a single neuron says, I'm going to calculate force. It doesn't know what torque is. And you know, people tried to come up with biological solutions for this quandary. You know, radial basis functions, gauge fields, and so on. People like Zipser, Teresinovsky, they tried to find a solution. But we understood all along that this was just a metaphor for what might be going on, and it might not necessarily really capture the underlying causal processes. However, I hold strongly that these empirically resolved response properties of single neurons and neural populations, and the differences in the response properties within and across regions, they may be coincidental, but they are not accidental. They can still provide some useful guidance in the different contributions that each area makes to the control of voluntary neurons. And there's a vast and rich body of literature out there describing the response properties of neurons and neural populations in multiple cortical areas that have been tested in a wide range and variety of test conditions that can provide inspiration for future studies using these new conceptual models and tools to provide deeper insight into the cortical neural mechanisms of voluntary motor control. And I'm thinking of literature, which is purely random. Let's say, for instance, my idea, how can we explain these correspondence between these simulations and the response properties of primary motor cortex neurons? How does the motor cortex deal with the anisotropic biomechanics of the limb, while the limb is trying to make movements in Cartesian space? How does the motor cortex know that there's an external load, and how does it know how to compensate for the external load? These are important questions. How do these differences arise between the properties of single neurons in the motor cortex versus the frontal cortex, both at the single neuron level and at the population level? How can we account for these different temporal dynamics of area 5 neurons versus area 4 neurons? How can we account for the fact that area motor cortex neurons are very sensitive to the posture of the limb, while premotor cortex neurons seem to be much less sensitive to the posture of the limb? How can we explain these striking differences in activity of neurons in the premotor cortex versus primary motor cortex when the animal's making movements either the contralateral or ipsilateral limb? You know, do these different response properties represent different levels of representation of intended motor outputs in different reference frames, such as, you know, in the metaphor of the sensory motor coordinate transformation model? Are these somehow different factors in an optimal control policy? Could these be different components of an unfolding neural trajectory in output null and then output potent parts of neural activity space? Choose whatever and flavor of the month model, semantics, and metaphors and choose. We have to have a way to explain these strikingly different empirically observed properties of neurons in different areas and different task conditions. So here's some thoughts I have about maybe where we should go in the future. I think it's very useful and important to compare the response properties of neural populations recorded simultaneously in different cortical areas and to apply these new tools, not just to analyze the activity within each area independently, but somehow to pull together all of these activities in different areas and extract from them some insight into how these neural populations interact in real time to produce these empirically observed ranges of response properties that we've seen in different areas and in different tasks. To date, all of the studies have focused almost exclusively on how motor cortical areas generate descending motor commands to control muscle activity. And I think we have to expand these studies to start dealing with higher order issues of planning, of action selection, and other aspects of action-related decision making. And ultimately, we still need to understand how the motor system integrates multiple sources of peripheral sensory and central signals to select and implement a plan of action and to execute the specific movements needed to fulfill some beneficial goal, which was the original motivation for the now disgraced sensory motor coordination transformation model. We need to get a handle on these issues using these new tools and new technology. So, I've often said that perhaps my most proud accomplishment is the people I've trained in my lab. I've never had a large lab. I couldn't manage a big lab. I couldn't remember all their names. So I had a large lab. But people have survived the experience and have gone on, many of them have gone on to successful careers in academics. Others came to their senses and got real jobs in places like, in areas like medicine, in other professions, and even in private enterprise. And of course, my lasting gratitude, debt of gratitude to the people who have supported our research over the years, in particular the CIHRP, Institutes of Health Research, and other sources of funding over the years. I've gone on pretty long. I wonder if you can indulge me a few more minutes, because there's some new data I would love to show you. I don't want to just dwell on my faded past glory. I'd like to show you some new data. I'm going to skip a lot of preliminary background stuff, okay? Maybe just get to the nub of things. We've been recording in a task in which monkeys make decisions based on a checkerboard stimulus in which we can manipulate the amount of color information. So the animal has to, again, choose between two color-coded targets, and based on his estimate of what's the dominant color of the checkerboard, point to the corresponding target. And we found that Emily Coyer and Christine Montanet have shown that premotor neurons show rapid changes in activity when the stimuli are easy and gradual buildup of activity when the stimuli are harder to make the decisions. More blue, more yellow, therefore she pointed out there's a blue-yellow target. There's a problem with this paradigm. There's a conflict. How do we interpret this presumably decision-related activity? Because in theory, you can identify at least two different kinds of decisions that the monkey is making. He has to make a perceptual decision as to what is the dominant color of the checkerboard, because that's the critical clue that tells him in which direction he should point, to which target he should point. And of course, there's also the action-related decision. I should point left or right, based on that sensory information. So we introduced a new paradigm a few years ago, in which rather than showing them the colored targets first and then the checkerboard, we showed them the checkerboard first and then the colored targets. So in theory, that should allow the subjects to look at the checkerboard, make their perceptual decisions in more blue and yellow, and then wait until the targets appear and then point to the corresponding target. So we're attempting to dissociate in time the perceptual aspects of the task from the action-related aspects. And this is a clearer picture the types of stimuli we use. Emily Croyer did a study in humans to look at how humans behave. These are cumulative frequency histograms of the reaction times of neurons when the stimuli are easy and progressively harder. So it takes longer and longer to make up your mind. And at the same time, you make more and more errors in your choices as the stimuli get harder. Just exactly a classic result for these kinds of perceptual decision-making paradigms. And in fact, you can model this behavior by a simple group diffusion model. However, when we showed the subjects a checkerboard first, the reaction time distributions changed dramatically. They became much faster than the checkerboard appeared. So clearly, they have made up their mind about what the dominant color is, and they're much more ready to point to the correct target. There is still some hesitation when the stimulus is harder than when it's easy. But the paradigm accomplishes its basic objective. We try to dissociate in time the perceptual from the action-related decisions. Now, Cristeva Montanet has now followed up those human studies with monkey studies. These are these two tasks, what we'll call the target first task and the checkerboard task first task. We use a smaller range of stimuli for practical reasons. And her monkey shows just what the humans do. There's a range of reaction times when the stimuli are easy, intermediate, and hard. And when he's looked at the checkerboard for a while before we show the targets, we see this collapse of reaction times, just as the humans had shown. So this is a sign that that monkey had made his perceptual decision while looking at the checkerboard and waiting for the targets to appear. Now, about the time we got to this, Cristeva had trained her monkey to implant a chamber over the motor cortex and premotor cortex started. We discovered that a trainee in Krishna Shenoy's lab, Megan Wang, was doing essentially the same experiment. So we decided to get together, pool our data, and publish a joint paper. These are the results. This is Megan's monkey. This is Cristeva's monkey on the right. In the targets first task, and then the checkerboard first task here. The checkerboards appear here, and then the targets appear here, and then the checkerboard. This was a take-home message for that experiment. We did a linear regression analysis of the activity of single neurons in 20 millisecond slices of time from the start to the end of the trial. And we did a regression against 4 predictors. The direction of the chosen target, irrespective of what the color was, that's the green line. The color of the chosen target, irrespective of the spatial location of the target, that's the magenta line. And also against the strength of the evidence in the stimulus favoring a particular spatial location of the target, independent of its color, and the strength of the color evidence in the checkerboard, independent of where the targets were. And as you can see, these cells do very little while the monkey is looking at those first stimuli, the entire task. Even though we have good reason to believe the monkey is looking at the checkerboard, making up his mind as more blue or yellow, premotor cortex cells showed nothing. So very little during this period of observation of checkerboard. They only become active when the second stimulus appears, be that the checkerboard or the 2 targets. And the activity strongly reflects the chosen direction of the target independent of its color. This is the proportion of cells that show statistically significant correlation of that particular in the linear regression. Many cells also show a correlation with the strength of the evidence that the monkey had made his decision. And essentially no neuron showed a correlation either with the color of the chosen target or the strength of the color dominance of this checkerboard stimuli. So all these things suggest that premotor cells are not particularly or at all implicated in the perceptual components of the task. They're mainly interested in processing information from these stimuli that tell the animal information about the spatial directionality of the motor output. So this strongly argues that premotor cells in this paradigm are mainly action-related, not involved in perceptual aspects of the task. This paper just came out, so you can all rush out and read it afterwards. But there's something interesting. Notice how these cells, as I said, are coding information about the strength of evidence that the animal used to base a decision, both prior to movement, during movement, and even while he's holding his arm out of the peripheral target. So why is that? What's going on? So let's think about what is going on in this paradigm. The monkey is, we'll just look at the targets first. We show him two targets, we give him a checkerboard, he has to use that information to choose which is the correct target, let's say it's blue. He points to the blue target, he waits for a little while, and then he gets knowledge of the results. If he made a correct choice, he gets a few drops of juice and he uses the click of the solar board. If he happens to make a wrong choice and goes in the other direction, we show him what the correct target was, and he gets no juice, no click of the solar board, no feedback. And he's making these choices on the basis of stimuli that are either very easy or much more difficult. This is a summary of his choice behavior as a function of the strength of the evidence, easy, intermediate, and hard stimuli. The red line is his reaction times. When the stimulus is easy, as we saw earlier on, he makes up his mind very quickly. The purple line is his success rate. As the stimulus gets harder and harder, it takes longer and longer to make his choice. And at the same time, in parallel, the likelihood that he made a correct choice diminishes. Now, the monkey has done this task 1 million times, 1 billion times, right? So when he sees a hard stimulus, he knows that there's a good chance about 1/3 of the time it's going to make an incorrect choice and not be rewarded. Even though he's investing much more time in making up his mind. So the next figure shows some other aspect, a different aspect of this video, what we call change in each direction. Sometimes a monkey starts to reach a target and then changes his mind and goes to the other target. He does this very infrequently when the stimulus is easy, and he does it more and more often as the stimulus gets harder and harder. About 6% of the time, he changes his mind when the stimulus is the hardest. Interestingly, these changes of mind or changes of direction correct initial errors, choice errors, more often than it actually cancels a correct choice, and the monkey points in the right direction. Finally, let's look at now velocities. These are 3 velocity curves of the pointing movements toward the target in trials in which he did not make a correction or change of mind. They're perfectly overlapping. So independent of the stress, the strength of the evidence the monkey made his choice, he moves to the target as very briskly in all 3 conditions. But look at what he's doing at the end of the trial. He's waiting for his reward to be delivered. If the stimulus was easy, this dark one, he actually starts to move back to the center before he gets his reward. Because he's sure he's going to get rewarded. He's just anticipating starting the next trial. As the stimulus gets harder and harder with his paler lines, he tends to linger longer and longer over the target, waiting to find out if he was rewarded or not. And if he makes a mistake, shown by this pink line, which means he actually pointed in the wrong direction, other direction, he lingers even longer, waiting to find out if he's going to get rewarded. And eventually recognizes, no, I made a mistake, I'm not going to get rewarded, and he goes back. So in summary, as the stimuli get harder, the monkey takes longer to make up his mind, he makes more errors, he changes his mind more often, and he lingers longer and longer at the end of the trial to find out whether he gets a reward or not. So all of these are behavioral signs that the animal is showing different degrees of confidence in his choices as a function of the strength of the evidence on which he's based the decision, i.e. metacognition. Again, remember, many cells show that activity reflects the strength of evidence both prior to, during, and after the movement. So let's look at some specific examples. Here's a neuron in those 2 tasks I told you about, target search and checkerboard first, aligned to the onset of the movement. The monkey is making movements at identical velocity to the target, but when the choice was easy, his activity was low, and as the choice got harder and harder, the activity increased. Kinematics are identical, and now he's holding over the outer targets, and we still see this expression of activity reflecting not only where his arm is, but how strong the evidence was on which he based this choice. There's another neuron showing the same phenomenon, but it alters signs. There's a third example of a neuron that shows a little bit of this effect of the strength of evidence prior to movement. And look what it's doing when he's holding over the two targets. He's holding his arm in two opposite spatial locations. He knows he has to move in two opposite directions to return to the center to start the next trial. But the activity is high when he had made his choice on the basis of an easy stimulus, lower with an intermediate stimulus, and lower still with a harder stimulus. Now I'm going to point out one very small little detail. Look at this little bump here in that pale line. Shortly after he gets his reward, after making a decision based on the weakest evidence, there's a little blip of activity. Right after he got his reward. That blip of activity here is not evident when he makes a mistake, the frown he faces, and he's not rewarded. There's another neuron again showing this modulation of activity based on the strength of evidence during and after movements. And again, subtly you can see that little blip of activity with the paler lines, the weaker strengths of evidence, not evident when he was making a decision on the basis of strong evidence, the dark line. And there's that blip again for the weakest evidence trials and correct choices. Look what happens when he makes a mistake. There's a abrupt, rapid decrease of activity and then some kind of a rebound. Clear difference in activity after correct trials and error trials. A bunch of other examples that are really quite striking. Look at this. Strong activity and rapid activity was waiting to be rewarded after an easy trial. When a trial is hard, the activity is suppressed. When he gets rewarded, there's this enormous burst of activity. Graded burst of activity is a function of the strength of the evidence in which you base this trials. And that burst of activity in correct trials is absent in error trials. There are four more examples of this phenomenon of graded activity after the rewards that modulated the function of the strength of evidence of the monkey's basis decision, and different responses when he had made a mistake. This now is the population activity, the mean population activity of the cells. And again, we see some modulation of the function of strength of evidence. It's much smaller at the population level because the activity is so different in single neurons. Sometimes they go up, sometimes they go down, sometimes it's only in preferred direction, sometimes in the opposite direction, sometimes in both. But we see the subtle effect. And this little burst right after the rewards. Let's zoom in on that burst. Here we show it here. When the animal has made a decision based on strong evidence, that burst is almost absent. When he makes decisions on the basis of weaker evidence, that burst gets stronger and stronger. Use the analogy, that burst, that post-war burst for only the weakest evidence trials. When he makes a mistake, he gets action. The cells transiently suppressed. I mean, these are all the canonical signs of a reward prediction error. But the reward prediction error here is not whether the reward is present or absent. He made a correct choice in every trial. The reward prediction error is based on how confident he was that he had made the right choice. When he was sure that he made the right choice, and he was sure he was going to get a reward, there's no burst. As the stimulus gets harder and harder, and so the likelihood that he'll get a reward gradually decreases, his confidence is lower, that burst gets stronger and stronger. So premotor cortex neurons are showing a lot of things going on in this trial that reflect the strength of evidence and a reward prediction error signal after the reward is delivered. And again, this discovery was entirely an accident. We saw this effect of graded activity during the movements, and I thought it would be a really good idea if we could change the data collection routine so we could look at what happens after the trial. Does something go on? And I mentioned this in my program, and looked at, you moron, you've been collecting that for 20 years. He had written debugging code in the software that continued to collect data for 1 second after the reward was delivered. We never looked at it. So those data are always there. We just went back and data mined and found this fascinating thing. So premotor cortex seemed to be involved not just in choosing actions, but also in the metacognitive process of assessing whether or not the animal is sure about his choices, and then showing signs of this reward prediction. I'm very sorry I went on so long, well past my allotted time. And thank you very much.